Hello students, today we are going to start a new chapter, that is free radicals and antioxidants. These free radicals are produced maybe because of this oxygen. This oxygen supply, which is very essential for us, could be a toxic when in excess. How it can be a toxic, that we will see, because it is converted into free radicals. Now what are these free radicals? A molecule or a molecular species that contains one or more unpaired electrons and is capable of independent existence are known as free radical. That is OH minus, O2 minus, all these charged particles which are known as free radicals. They are harmful to our biological body. Now we see an example, that this molecular oxygen, which is very essential for us, when it takes an electron, maybe this electron came from environment or from mitochondrial electron transport system or anywhere, then it gets converted into superoxide ion. This superoxide ions, when again it takes two hydrogen protons and it's get converted into H2O2. This H2O2 is not a free radical, but though it is highly reactive and hence known as reactive oxygen species. These are also dangerous or harmful to us. They are comparatively very active as free radicals. This hydrogen peroxide is converted into, you see here, hydroxyl radical, that is OH minus. In this way we see, with this molecular oxygen, many such reactive oxygen species are produced which are harmful to us. Now we see sources of or generation of free radicals, from where these free radicals are produced. These sources are of two types, that is cellular, by cellular metabolism, and by environmental effects. By cellular metabolism, it is from leakage of electron from electron transport chain, production of H2O2 or superoxide, that is O2 minus ion, by oxidase enzyme, that is xanthine oxidase and NADPH oxidase enzyme. They produce H2O2, that is reactive oxygen species. They are also produced by lipid peroxidation. When there is lipid peroxidation of a membrane, that is lipid membrane, when this membrane is converted into chain reactions of lipid peroxidation, then it produces free radicals. These free radicals are also produced by peroxisomal generation of H2O2 and superoxide ion. During the synthesis of prostaglandins, when the prostaglandins are synthesized, then it can also produce free radical. Then nitric oxide is produced from arginine amino acid. During the course of phagocytosis, when there is phagocytosis, that is when foreign particles or foreign molecules comes inside the cell and cell fights against it, that is phagocytosis, that forms free radical formation. In this, for phagocytosis, NADPH oxidase enzyme is used and that produces free radical formation. Now we see what are the environmental effects as a result of drug metabolism. So whatever the drugs or medicines we take may produce free radicals. Due to damage caused by ionizing radiations, even by the pollutions, air pollutions, air pollutants, that can also produce free radicals. Photolysis of oxygen, cigarette smoke also causes free radical, which is very harmful to our respiratory system. Then alcohol also produces free radical formation. Now generation of this ROS, reactive oxygen species, by macrophages. What we told about here, that phagocytosis. So during this phagocytosis, inflammatory cells, particularly macrophages, they produce superoxide ion by a reaction that is catalyzed by NADH oxidase system, which produces free radicals. This superoxide is converted to then again into H2O2 and then into hypochlorous acid by bacterial actions. So these all are harmful to us. Now characteristics of free radicals. Now what are the characteristics of free radical? Why they are harmful to us? They are highly reactive, they have very short half-life, they can generate new radicals by chain reaction. They causes chain reaction, that is one radical produces another, another will again produces third radical, third, fourth, or many such radicals, and in this way the chain reaction is formed and all these radicals are harmful. It can cause damage to biomolecules. Biomolecules means carbohydrate, lipid, proteins, nucleic acids, DNA, RNA, that is all these are damaged by these free radicals. Even the cells and tissues are also damaged. That's why these free radicals are harmful. These are the characteristics of free radicals. Now we see this excess free radical. It produces, you see, all types of disease, that is diabetes, cancer, gout, asthma, uric acid formation, arthritis, allergies, aging effect, cholesterol synthesis, that is atherosclerosis, insomnia, all are these caused by free radicals. So we see therefore free radical shows many harmful effects in our life. Now we see what are the harmful effects of free radicals. Free radicals and biomolecules, we have seen that they are very reactive, hence they damage all types of biomolecules. ROS, that is reactive oxygen species, they damage proteins by fragmentation, by cross-linking and by aggregation. As a result, they lose their biological activity. So proteins, after the free radical attack, they lose their biological activity. Lipids, we have I have repeated many times that these lipid are present in the membrane and they are lipid peroxides. They are peroxidation reaction takes place and these lipids are oxidized into lipid peroxides. These lipids, mainly PUFA, polyunsaturated fatty acids, are highly susceptible to this damage and causes the chain reactions. These carbohydrates, how carbohydrates are damaged? The oxidation of monosaccharides, they produce H2O2, that is reactive oxygen species, and also oxoaldehydes. Linking of this carbohydrate to protein, this carbohydrate when linked to protein are more susceptible, they cause proteins to be more attacked, or they are increase the susceptibility of proteins to attack by free radical damage. This free radical may cause DNA strand break, fragmentation of bases and deoxyribose. These changes causes mutation and cytotoxicity. So all these changes in the DNA strand causes mutation and cytotoxicity. Now free radical and disease. Now we see how free radical are damaged and causes disease. So free radicals have been implicated in causation and progress of many diseases. A cardiovascular disease, we see oxidized LDL, which produced by free radical, causes atherosclerosis. Free radicals damage DNA and causes mutagenicity and cytotoxicity and plays key role in carcinogenesis. In inflammatory disease like rheumatoid arthritis, chronic glomerulonephritis, ulcerative colitis, these all are caused by free radicals. How these are caused? Rheumatoid arthritis, it is self-propagated by free radicals and released by neutrophils. And neutrophils are released and causes free radical damage. Then we come to the Respiratory distress syndrome. This type respiratory distress syndrome, it is a disorder characterized by pulmonary edema that is also caused by free radical formation. Diabetes due to destruction of islets of pancreas may cause insulin deficiency and causes diabetes. Cataract formation, we see it is due to photochemical generation. It is due to photochemical generation of free radicals. Tissues of eyes, including the lens, have high concentration of free radical and scavenging enzymes, and that's why this free radical scavenging enzymes, they cause cataract to, that's why it is proved that cataract is formed by free radical attack, because the free radical damaging enzyme activity is increased there. That's why we can see that free radicals have been increased there. Such male infertility, aging process, in the aging process also free radical damage is seen. It plays in aging process, free radicals plays an important role and aggravate certain disease processes like diabetes mellitus, atherosclerosis, cancer, etc., liver cirrhosis. All these are because of aging processes and causes due to free radical attack. Reperfusion injury, that is also one of that is also caused by free radical attack, by as we see that myocardial ischemia is caused by free radical. During ischemia, the activity of xanthine oxidase is increased and hence free radical attack is increased. Now we see what is lipid peroxidation. Now what is lipid peroxidation? It is a chain reaction. It is a chain reaction that is initiated by free radical, which provide a continuous supply of other free radicals formed from unsaturated fatty acid, which initiate further peroxidation. That is, a one radical produces the another radical and another will produce the next radical. In this way they initiate further peroxidation. This type of lipid membrane destruction is known as lipid peroxidation. Now the stages of lipid peroxidation. Lipid peroxidation process takes place in 3 phases. The first one is initiation, second is propagation, and third is termination. So first one is initiation process, second is propagation process, and third is termination process. Now this initiation process, what happens? The polyunsaturated fatty acid, this is PUFA, present in the cell membrane are easily destroyed by peroxidation. During initiation phase, primary event is that the production of lipid radical. This production of lipid radical or ROO peroxyl radical, this R peroxyl radical, these both are produced in initiation phase. This R lipid radical and ROO radical, they are then degraded to form MDA, that we will see in the next slide. Now the number 2 stage is known as propagation stage. Propagation stage means, you know, elongation. How this chain reaction is converted. Now the lipid peroxidation chain reaction occurs. This occurs how? The free radical, this lipid free radical that is formed, this reacts with molecular oxygen to form peroxyl radical, which can attack another lipid or polyunsaturated fatty acid, that is lipid hydrocarbon, and converts it into lipid free radicals. So you see, after one free radical that produces this free radical and again one lipid free radical. These peroxides are capable of further stimulating lipid peroxidation, and in this way alkoxy and peroxy radicals are formed. These 2 radicals are more formed, and in this way many free radicals are formed and chain reaction takes place. As a result of these reactions, the conversion of lipid radical and peroxy radical, which would continuously produce hydroperoxides and produce other free radicals, and chain reaction takes place. Accumulation of such lipid damages lead to the destruction of fine architecture. In this way, they destroy this fine architecture of the membrane and integrity of this lipid membrane. So the integrity is also destroyed and the fine structure of this lipid is also destroyed by this lipid peroxidation. Now the third phase comes is termination phase. This lipid peroxidation proceeds as a chain reaction until all puff are oxidized. They go on and the reaction would proceed unchecked till a peroxyl radical reacts with another peroxyl radical. This peroxyl radical reacts with another peroxyl radical to form inactive products, and then only this lipid peroxidation will stop. Now this lipid peroxidation are estimated. How they are estimated? So they are estimated by MDA, that is Malondialdehyde, which is a marker. So by the initiation of lipid peroxidation, a product is produced that is known as MDA, which is the marker for lipid peroxidation. This MDA is then estimated by thiobarbituric acid reactive substances, and they give red color substances. In this way they are estimated. The products of lipid peroxidation are highly destructive. They damage membrane, they lose their integrity of the membrane, they even destroy tissues and cells. Now we come to next after the free radical, that is antioxidant. So what are these antioxidants? You all know that free radicals are very harmful, but God have created some defense against these free radicals, and this defense present in our body are known as antioxidants. These antioxidants are substances which scavenge free radical and stop oxidation, hence they are known as antioxidants. Now we see the classification of antioxidant. So antioxidation in presence of lipid peroxidation, as according to the lipid peroxidation, these antioxidants are classified into two antioxidants: preventive antioxidant and chain breaking antioxidant. Those antioxidants which break the chain of lipid peroxidation, they are known as chain breaking antioxidants. They are one enzyme, that is superoxide dismutase enzyme. Vitamin E also is a chain breaking antioxidant, and the other is uric acid. And preventive antioxidant which prevent this lipid peroxidation are catalase enzyme and glutathione peroxidase enzyme. Now antioxidant, according to their location where they are present, on that basis these antioxidants are divided into three. That is, the antioxidants present in plasma are plasma antioxidants, present in cell membrane are cell membrane antioxidant, intracellular antioxidants which are present in intracellular. So in plasma, vitamin C, beta carotene, bilirubin, uric acid, ceruloplasmin, transferrin, all are examples of plasma antioxidant. Even in the cell membrane which are present, we have already seen that PUFA in the lipid membrane, they are chain breaking antioxidant, that is alpha tocopherol, that is vitamin E. Then intracellular, inside the cell are present that are SOD, that is superoxide dismutase, which act on superoxide ion, catalase which act on H2O2, that is hydrogen peroxide, and glutathione peroxidase, that also acts on peroxidase H2O2. Now antioxidant, according to their nature and action. According to their nature and action, antioxidants are classified into two. That is, when they act as enzymatically the catalytic reaction, then they are enzymatic antioxidant. When they don't do any that type of work, or action, they are non-enzymatic. Enzymatic and antioxidants are all the enzymes present in our cells, that is catalase, glutathione peroxidase, superoxide dismutase, glutathione reductase. And in non-enzymatic, again they are divided into nutrient and metabolic antioxidant. Nutrient antioxidant which we take in our diet are beta carotene, alpha tocopherol, ascorbic acid, selenium. These are all nutrient antioxidant. Then we come to the metabolic antioxidant, which are produced as a metabolism in our body, like glutathione, which is very essential antioxidant present in RBC membrane. So RBC membrane is very prone to oxygen. So it gets this glutathione is always in reduced form, which takes H2O2 produced in RBC membrane and get oxidized, and this again by the NADH which produces this reduced glutathione. In this way it protects the RBC membrane. Then ceruloplasmin, albumin, bilirubin, transferrin, ferritin, uric acid, these all are again metabolic antioxidants. Now we see some of the antioxidant enzyme systems, that is superoxide dismutase enzyme, that is known as SOD. It acts on superoxide ion. Okay? This is O2 minus, here there is a mistake. So it converts this superoxide ion, that is O2 minus, into H2O2. It produces this H2O2, and this in the presence of enzyme superoxide dismutase. So it reduces the effect of this superoxide ion. This is the first line of defense to protect cells from the injurious effects of peroxides. Then the second enzyme is catalase, which act on this hydrogen peroxide and gets converted into water and reduces the effect of hydrogen peroxide. Then the third enzyme is glutathione peroxidase. This glutathione peroxidase, it acts on hydrogen peroxide and gets converted into water. This glutathione, it converts this glutathione into oxidized. This reduced glutathione takes H from this H2O2 and gets converted into oxidized glutathione. Okay? H2O2, it takes H and gets converted into oxidized, it takes oxygen and gets converted into oxidized glutathione. This oxidized glutathione is again converted into reduced glutathione in presence of glutathione reductase. This glutathione reductase plays important role. It converts this NADPH into NADPH. This NADP into NADPH. This hydrogen is then given to this oxidized glutathione. This gets converted into now reduced glutathione. Now next is tocopherol, that is alpha tocopherol, which is vitamin E. It is present in cellular membrane and protects cell against chain breaking antioxidant. It is chain breaking antioxidant. It breaks the lipid peroxidation. Now we come next antioxidants, that is ascorbic acid. This is vitamin C. It participates in many normal reactions. It is important water soluble antioxidant. It scavenges free radical and inhibit lipid peroxidation. It helps vitamin A in lipid peroxidation. It promotes regeneration of alpha tocopherol, that is vitamin E. Then comes beta carotene. It is antioxidant under low partial pressure of oxygen. It functions in association with vitamin E and vitamin C. Lycopene, a fat soluble pigment, is a carotenoid responsible for color of the certain fruits. Lutein and xanthine impart yellow color or green color to the fruits and vegetables. So all the colorful fruits and vegetables, they are antioxidant products. So we have to improve our diet with all these colorful substances. Even in our Indian kitchen, all the spices what we use, they are all act as antioxidants. Now we see many sources of what nutrient antioxidant, like vitamin E. It is present in unprocessed vitamin oils, whole grain, leafy vegetables, legumes, etc. In vitamin C are present in citrus fruit, cauliflower, melons. Beta carotene is present in carrots, green fruits, vegetables, spinach, turnips, apricots. Lycopene is present in tomatoes and other products. Papaya, pink, guava, watermelon, all these contain lycopene. Selenium is present in seafoods, meats, organ meats, whole grain, and catechins are present in green tea. That's why nowadays everybody have started to drink green tea rather than that milk tea. Curcuminoids, it is present in turmeric, that is also a very good antioxidant. Now thank you very much. So